We will move to the very first presentation, and it is my greatest pleasure to introduce to you all Dr. Lucy Woodall. Lucy leads the scientific research aspect of Nekton and is also a senior fellow, research fellow at the University of Oxford. She's a marine biologist with strong passion for sharing the delights and importance of the aquatic world with global citizens. Whilst her work broadly focuses on the processes that drive biodiversity in the ocean, she's also interested in understanding the impacts of human activities on these ecosystems. And she's a member of many applied working groups, such as the high-level panel expert group for the UN on sustainable ocean economy, the IUCN SSC Seahorse, Pipefish and Seadragon specialist group, and co-leads the DOSI pollution and debris working group. So it's my pleasure to introduce you to Dr. Woodall, who will be talking about the Nekton science objectives and general overview. So Dr. Woodall, on to you.
Thank you so much, PS. I'm just going to share my screen. Okay. So I have the absolute honor of starting this session today. And what I'm going to do is try and set the scene of the Seychelles First Descent Expedition, which is one of the many fantastic projects that are going to be shared this week. And of course, the samples and data collected from this expedition are going to be used by the rest of the speakers today. However, before I go any further, I just want to say thank you to the groups and the individuals responsible for putting this event on. I'm super looking forward to it. So the first Descent Seychelles expedition was conducted in March and April of 2019. And you can see kind of this timeline of activities. Because as all of us who head into the field know, research doesn't start when we don our snorkels. And in this case, the expedition started in 2018, when we established and then had multiple dialogues with fantastic partners. As you can see from this schematic, the field phase is just one of many steps that is required to ensure that desired data are collected and then that is turned into usable information. Today, the team is going to be sharing some of this information, and we all really look forward to discussions with everyone who's able to attend this symposium about the relevance and the interpretations of these data. I'm going to mainly highlight the at-sea activities in my talk today, but of course, if you're interested in the other phases, then please do get in touch. So this project has 3 pillars or parts. The first was to provide new information and learning from the science, to share this knowledge and skills, and to communicate the value and the awesomeness of the ocean, especially the deep reefs. The science objectives were co-defined, and then the science outputs are now being co-delivered with many of the people participating this week. And of course, many others too numerous to mention now. The overarching goal of the research really was to establish a baseline of ocean life and conditions. That means the biology and the environmental parameters. And together, these data will contribute information to the marine management plans of the newly established MPAs in Seychelles, and also to help us better understand the patterns of life in the waters, especially the deep reefs. So our multidisciplinary research focused on slope ecosystems down to 300 meters. And these data connect the fantastic studies that others are doing in shallow waters, of course, some of which we'll hear about this week, with deeper water observations, bringing together these two aspects. As I've already shared, many people were part of this project, and notably, this included projects co-funded by Nekton and SACAT, providing grants and leadership for Seychelles scientists to conduct deeper ocean studies. And also a fellowship program jointly funded by Nekton and AFOX, which is the Africa Oxford Initiative, which provided visits to Oxford University for 2 grantees. We've also conducted taxonomic workshop, skills training workshops, and are just starting a journal club. And this is to share expertise and maintain a network of like-minded researchers. And public engagement was there to, and is still being used to create inspirational content and stories. The expedition was joined by Sky, who were also able to cover the Aldabra cleanup project, and we'll hear more about that later this week, and also joined by Associated Press. Together, they provided worldwide coverage of the expedition and continue to be supportive of this work. Tomorrow, there's going to be a broadcast of an updated documentary on SBC. So many of the speakers and organisers of this symposium have starring roles. So from the planning prior and during the expedition, we were able to visit 7 different sites, covering over 2,000 nautical miles around the outer islands of Seychelles. And you can see from the map here that our first site was in Alphonse, then we moved to 2 sites in Aldabra, and then to Asto before leaving the Aldabra group of islands back to the Amarantes, doing some more work in Astove, and then Poivre, St. Joseph, and Desroches. Next to the vessel, she was called the Ocean Zephyr, and she was chartered from the commercial fleet and had previously been used as a platform support vessel. As she wasn't a dedicated science vessel, she did require adaptations for the communications and the science activities. I'm not going to detail the communications activities and aspects here, but of course they were many, but instead I'm just going to share the equipment that we used for science. So we had the submersibles that allowed us to survey and collect data, and of course to sample some organisms. We had remotely operated vehicles that we used on surveys, a Neuston net for collecting surface zooplankton samples, and a multi-beam on this mapping boat to collect bathymetry. We deployed a CTD to measure conductivity, temperature, and depth, conductivity being a proxy for salinity. And we also had a number of other sensors to collect parameters of the water column, like light and oxygen. We deployed a drop camera and a hydrophone on occasion as well. We're also able to integrate an ADCP to measure ocean currents, a USB-L system to detect and record positions of deployed gear, and also an underwater telephone to communicate with the submersibles. Finally, on deck, there were support craft. There were containers to store samples and also to use as labs. And the communications team had media rooms, a live broadcast link, mission control, satellite dishes, and live cameras. So now just looking at the submersibles themselves, sampling was done by Omega. You can see here that she had an arm and a box for collecting megafauna. And sampling was only done to ground truth video and describe new species, as well as to document diversity. She was able to sample some water, and these are the Niskin bottles and the CTD sensor. And finally, she had an HD camera and lights on the side. And this was used to document organisms that were collected in situ, and they're specifically useful for the field ID guide. Kensington Deep was the survey submersible. Again, she had powerful lights that you can see on the side. And we utilized a stereo video system. And the stereo system means that we can't just count things, but we can also measure the length and the area. And this means that we can give quantitative measures of surface area, but also biomass when we look at fish. So we had cameras that pointed down for the benthos, forward for the demersal fish communities, and then we also had cameras on the side, and that's what you can see here. The cameras have the little blue ends. You can see this pointing out to the side, and this was to capture the slope of the seamounts, because in some areas that we'll learn later today, they were particularly steep. So what did all of this mean? We were able to conduct deployments 219 times of a whole range of different scientific equipment, from ROVs through to hydrophones and scuba dives. There were 209 specific megafauna specimens that were collected. And then these resulted in over 2,000 samples, which were either the epifauna or the megafauna samples, like you can see in the background slide here, the little brittle stars. They were some of the samples of the nets and subsamples of the megafauna as well, that were destined for different activities. And we're also able to conduct 209 different video transects from across the 7 different locations and multiple depths. So I'm going to finish as I started by thanking everyone. And of course, there are so many. And thank you all for your valuable contributions. You can see in this slide just a few of those who were involved in the expedition. But without everyone, this work would not have been possible. And this really was the work of partnerships. And not only thank you for being involved and for contributing, but thank you for making this project not just successful, but a joy. And thanks very much, everyone.
Thank you so much, Dr. Woodall. It brings back so many memories. I had the best of luck of being part of the expedition. So it was a chance and experience of a lifetime, and certainly great things, slowly are coming out from the expedition. So thank you very much once again. We're just checking on the questions. We have 2 questions. The first one is, are there any plans for any future expeditions in Seychelles? And secondly, if Dr. Woodall, you can share preliminary details of any new species found during the expedition.
Fantastic. Thank you so much for those questions. Yes, so let me answer the first one first about expeditions. Of course, some of you might know that we were in Seychelles trying to undergo another expedition when really this awful global pandemic of COVID hit, and we had to postpone that expedition. And the plan there was for us to think about understanding some of the slope habitats in slightly deeper waters in the open seas. And that meant we were going to try and explore some of the seamount communities, We still have aims to do that, and would love to hear from anyone who is particularly interested in that aspect. But exactly when, we can't say, but we really hope we're going to be back in the water next year. And I'm sure everybody's got fingers crossed that, you know, the global situation will allow us and others to continue with our field research. Regarding new species, then what I can say now is absolutely we have new species all the way from brittle stars through to different corals. I'm not going to give you exact details on those because we have speakers today who are the expert taxonomists in those particular taxa, and they will give you the details. But there are new species, and we're particularly delighted that we can share some of those findings with you all today.
Just to add on to what Lucy mentioned, especially for Frances, who sent the question about the new species, there is the small video which will be airing on SBC tonight, SBC1. I mean tomorrow evening, sorry. But however, I'm sure we'll have it on YouTube. So Frances, you will have the chance to see it online eventually, hopefully by the end of the week. Okay, so it seems we don't have any other questions for Dr. Woodall. So we have one more. Is there any way a person from Kenya can be integrated into the research projects, Nekton's research projects?
Thank you. Thanks for that question as well. So you bring up a really important point that science research done in Seychelles needs to be kind of sat within the whole of the region. And of course, this is incredibly important. And, you know, why our research doesn't necessarily just focus on Seychelles. It was a starting point to be able to branch out into other areas and to really understand the research within context. So we've got a number of initiatives right now, and we're just going through some agreements with partners, so I can't talk too much about them, but please just send me an email, whoever asked that question, and I'll be able to share some more details in the future. But we're really hoping that this is going to be a regional opportunity, and there will be opportunities for more in-depth discussions across the region. So thanks so much for that.
Okay, one last question before we move on to the next presentation. Will the collected data be available to the public so that other projects can go further in these specific fields?
Yeah, absolutely. So open data is a really important aspect for this expedition. And what we're doing now is just getting all the science teams working up these data. And then together with ministry, we're going to be looking at how best to share this information. But if you particularly have an interest in a particular type of data, then again, please get in touch. There's copies of the data with us in the UK, but obviously also in Seychelles. So it is accessible for specific questions right now, but it'll be open to a wider audience in the future.
Thank you very much. So we'll move on to the next presentation by Dr. Jim Mortimer on the seagrass ecosystems and the deep sea of the outer islands of Seychelles. So Dr. Mortimer is a conservation biologist, originally from the US, who has worked to promote conservation of endangered species and habitats, primarily sea turtles and tropical, coastal, and marine ecosystems around the world. Since 1973, she has worked in some 20 countries on 6 continents. Her interest in seagrass ecosystems began all the way back in 1975, while she was doing her MSc study on the feeding ecology of green turtles hunted by Mosquito Indians along the Caribbean coast of Nicaragua. She first came to Seychelles in 1981 and became a naturalized citizen in 2007. Since 1995, her geographic focus has been the Western Indian Ocean, especially Seychelles and the Chagos Archipelago. An additional information on Jin, she is locally known as Madame Torti, that translates as Mrs. Turtle in Seychelles. So that's how well known she is with everybody of all age groups in Seychelles. So on to you, Jane.